So I just want to point out, although like everything's been really focused on how complicated autoimmune is, dietary, nutrition, lifestyle approaches is really profound in many patients that have autoimmunity. So the key thing you want to understand about all the things we covered is that not that there's no hope to do anything for patients that have autoimmunity, there's definitely hope, and all the different genes and things we talked about can be expressed differently with different lifestyle, nutrition, dietary approaches. And these people that have autoimmunity have really nowhere to go. So if you don't help them, I mean, most of them are just lost. So, just know that, and know that the key thing about this morning wasn't that you can't do anything clinically with them. The key thing with this morning is you have to realize that they're all unique and different, and you have to have that level of openness and expect that diversity, with their clinical presentation, and expect that diversity with things that work for them and things that don't work for them. Then know that every single case is totally different. There are some patterns where you see a person that has chemical-exposed autoimmunity or antigen-exposed autoimmunity or just dysregulation or loss of tolerance, and those that just have endotoxemia that really are there, and you can kind of pinpoint those out or work through those. So that's hopeful, and you can do a lot of things for them. But the focus right now is to start getting into some of the clinical concepts, and we'll continue to go into the clinical application with this throughout the course. But let's talk about diet, and this is a critical thing, okay? So when we look at dietary proteins and autoimmunity, one of the things we need to know is that for the most part, people that have autoimmunity, they've lost tolerance. That's one of the key features of all the different autoimmune disease variations. They all fundamentally have lost tolerance. And tolerance is an immunological term about reactivity to either food proteins, which is called oral tolerance, chemicals, which is chemical tolerance, or to cell tissue, which is what autoimmunity is. It's losing tolerance to your own cell tissue. So you're going to expect pretty much every one of your autoimmune disease patients to have dietary protein intolerances because it's part of losing your tolerance. So they're going to have sensitivities and reactions to different types of food proteins. And what you want to try to distinguish is what kind of diet to put them on or what specific foods. So we're going to talk about general immune reactions and then cross-reactive reactions. And there's definitely people that cannot handle any lectins. Some people can't handle any nightshades. Some people can only do autoimmune paleo. Some people just need to be off gluten. Some people, you'll have to follow specific cross-reactive foods. There's some research about specific cross-reactive foods, about thyroid and type 1 diabetes and some other autoimmune diseases that I'll share with you that are more specific to the condition based on what's been published on it. But you're going to expect that you're going to have to make some dietary management with your autoimmune disease patients, okay? So where we are in the course right now is we've talked about the basic review of immunology and fundamental concepts. We talked about the landscape. Now we're going, okay, we have the landscape. Someone's come in, they have autoimmune disease. What kind of diet recommendation should we give them? Should we give them all gluten-free diet? Should we give them all autoimmune paleo? Should we be more or less restrictive? And these are things that you have to do in real practice. But also in real practice, you have different levels of patient motivation as well, okay? So we'll go over the literature regarding dietary proteins and autoimmunity, and then we'll talk about clinical skills of how to develop a dietary program and recommendations for patients with autoimmunity. And then there's several key concepts that I want to definitely cover as we go through. So number one, dietary proteins change their antigenicity if they're cooked or raw. That's a really important concept. Some people won't react to one type of version of a food versus another. So cooked and raw is the essential part. Dietary proteins can change their antigenicity when they're combined with other foods. So real-world food reactivity could be a combination of foods have a different response than isolated food proteins together. And then dietary proteins can cross-react with other food proteins. So this is where you have sensitivity to multiple foods because of the same amino acid sequence. And then dietary proteins cross-react with body tissue proteins. That's where a food protein that a person's sensitive to makes antibodies, and it actually attaches to their own tissue and promotes the autoimmune response. And then dietary proteins can induce immune reactivity by either specific cross-reactivity or direct by immune pathway activation. So let me just go over the basic concepts first, these 5 key concepts, then we'll move on to a lot of other material. So the key thing is dietary proteins change their antigenicity depending on whether they are cooked or raw. So the protein structure of food proteins can change antigenicity when cooked. Some patients will react differently to raw foods versus cooked foods. This was a diagram Dr. Vijdani made, and he just cooked eggs at different times just so you can actually visibly see the protein structures change. So when you look at protein structures, remember proteins have an amino acid sequence, and that amino acid sequence allows for different antibodies to bind to it. And when you cook, you change the structure. So you can have someone who reacts to raw tuna when they have sushi, but not to cooked tuna. Okay? So this was a video that I got that Cyrex made, but it goes into cooking. I got this illustrated for you. So basically, they're showing a protein structure there, and then with the protein structure, you can see when they cook it, you can actually see the sequence of amino acids, how they get exposed to the environment change. That's the concept of how heating changes a food protein. So when you're looking at what's been published on this, this is one of my favorite titles of a paper, Rare, Medium, or Well Done, the effect of heating on food matrix and food protein allergenicity. And they basically go on to say heating and other methods of food processing have different effects on food allergens and even those contained in the same complex food. So there was a study that Dr. Vijnani published, and here you can see different food proteins that he tested. So there's 160 subjects, and then you can see which percent of the subjects react to different food proteins. But take a look at these ones here. Of these 160 subjects, when they tested them against raw salmon, 28 showed up. When they checked cooked salmon, 126 showed up. Same exact protein, but just raw versus cooked. Here you can see raw shrimp, 25, and then fried shrimp, 134. Raw bacon, who eats that? And then fried bacon. But you guys are laughing, but let me explain something to you. When you do a food sensitivity test, it's always raw. So if you do raw, so you do Cyrex. And by the way, I work with Cyrex, and part of the reason I work with Cyrex is because they're the awesome lab that does everything autoimmune that's awesome. But one of the things that a lot of the research has shown, besides Dr. Vijnani's work, like the papers I showed you, is that they changed their antigenicity. So unless the lab is saying cooked versus raw, you're looking at raw. If it's cooked, they will actually list cooked. And it's very difficult to do. So I don't know if you guys have ever been in the lab to do any kind of antibody testing, but what you do is you get a well, and then you get proteins, and then you have to purify the proteins. And then so you have pure protein, you have to make sure there's no debris, and then you put those proteins on a lysa plate in these different wells, there's different things in there, and then you let it bind, and then you clean it out, and you let it bind, you clean it out, let it bind, so it's just pure protein stuck to this, and then you take the patient's blood, you put the blood in, and you see if the antibodies bind to it, and then you have to find the right degree of saturation of antigens that plate it, and it's different, like fried shrimp could have a 40% concentration, and raw tuna could have a 30% concentration. You have to do all that in the lab to figure that out. It's an extensive process. Most of the labs that do food sensitivity testing buy pre-made food protein wells that are all raw, and they have a little bit of protein in there, and they test for it. So when you guys see a patient that has pork test, for example, which is raw bacon, or raw beef, or what a beef or pork tested, they're measuring it raw. So you will, unless you see the word cooked exactly next to the food protein, it's not cooked. And changing the structure of the protein with heat can have an impact. Like again, who eats raw bacon? How many people eat raw egg? People eat cooked egg. And if you look at the degree of reactions in the sample population, you can see there's a dramatic difference between raw versus cooked foods. So in this paper here, published in Nutrition and Metabolism, commercial food antigens are prepared from raw food. Furthermore, all IgE and Ig antibodies against dietary proteins offered by medical laboratories are measured against raw food antigens. So that's the key thing you want to be aware of. Because at some point, if you have a person that has autoimmunity, you're going, what kind of foods am I going to have this person eat or not eat? Do I want to do food sensitivity testing? If I do food sensitivity testing, what am I really looking at? Okay? So the first thing you have to realize when you do food sensitivity testing is whether you're looking at raw or cooked. And clinically, they do have a difference in their outcomes. So dietary proteins can change your antigenicity when they're also combined with other food proteins, besides being raw and cooked. So take a look at these results here. Okay, so if you look at the ingredients of sausage, soy, raw meat, wheat, and the actual sausage, difference in reactions when they test the whole food protein, cooked, than when it was just the individual ingredients. Okay, take a look at here, fried chicken. Wheat has so many reactions, chicken has so many reactions, but when it's together, wheat and chicken combined, fried chicken, completely different antigenicity. You can take a look here. Individually ingredients for beer, but beer itself, or look at donut, wheat, barley, yeast, soy, but when they're mixed together as a donut, completely different. So they basically do is, in the study, they take donut, purified it into a protein, or if it already cooked mixed together, and then checked it against the actual individual ingredients, and there's a big difference. So real life reactions with food sensitivities are different than what you can see with a panel. Okay, so first of all, the key thing with looking at antibody food panels is, unless it says cooked, you're not looking at cooked, you're looking at raw. You're going to miss a lot of antigenicity only in raw forms. Okay, so I prefer the Cyrex Ray 10, and I have a financial relationship. I'm a consultant to Cyrex. It is by choice. I have other opportunities, but they do the best autoimmune stuff. And the reason I do it is because the person who runs all the tests is Dr. Vijdani, who's been my mentor my whole career. So he published some breakthrough stuff on raw versus cooked. So have other immunologists, and it's a real factor. And quite honestly, you have to. It takes a world-class lab to purify their own proteins in cooked and raw form and offer that in as a commercial test. You can't just open up a lab and do it. You have to be an expert. And I don't know if you guys know this about Dr. Vijdani, but Dr. Vijdani was the first person that did antibody testing out of UCLA for foods. So he's really the main laboratory guy 45 years ago that really did major studies on food sensitivity and autoimmunity and food reactions and allergy and food sensitivity versus allergy and all that stuff. So he basically has shown these types of reactions because as an immunologist, he knows that food protein in the real world, someone can eat a mixture of foods cooked together and have a significant reaction, but they didn't test for it. So the combining of real foods, okay? So what does that mean in testing scenarios? Should you test, should you not test? Yes, you should. You should test when you want to look for things because patients don't always have a clear symptom, but you want to know the limitations of it. And you should also not discount when a patient says, hey, I ate these foods, they were all on the list that were clean, but when I ate it, I totally reacted to it. So if someone eats, I don't know, Mr. Chang's Kung Fu Pao chicken that's gluten-free, doesn't have any ingredients, but they react to it, and they swear it's not contamination. Maybe it wasn't contamination. Maybe it was just the combining of those food proteins for them. So guess what? They can't eat that Kung Fu chicken or whatever it was that caused that reaction. So there is some trial and error to look at these things. So you definitely want to look for food sensitivities with your patients. Some of them, they can clearly feel. Some you don't know. And also, we'll talk about food sensitivity. You want to also run testing sometimes because you want to know, are they reactive to everything they're eating? Which means they've lost their total or oral tolerance, or are they just reacting to a few foods, which means their tolerance is intact. So food sensitivity testing can give you some clues about what's happening with their tolerance to the immune system. Is it everything or just a few things, okay? So know that patients can have their own unique interactions based on how foods are cooked, okay? And then they have to really listen to those and observe those as well. There's some more samples of these things, like a pizza, for example. All the individual ingredients besides, or just pizza by itself, okay? So, clinical pro. The antigenicity of individual dietary proteins can be changed when combined with other food proteins. Real-world combining of dietary proteins should be considered when interpreting reactions to foods. Okay, now, dietary proteins can cross-react with other food proteins. So, in the case of an immune reaction, if two food proteins share a similar sequence of amino acids, then antibodies made for one protein may bind to other similar amino acid sequences of other food proteins. So if you look over here, you see the amino acid sequence of a consumed food, and then here you see another food protein, but there's a similar amino acid sequence. You all see that here? Okay, so that's cross-reactivity. So this was a great paper published in Journal of Allergy and Clinical Immunology, and they went and they explained this concept. They said lots of food proteins in the same group have amino acid sequences that are similar. So he said if a person reacts to salmon, 50% of them will react to other fish because amino acids are similar against fish species. If a person reacts to cow's milk, 92% of them will react to goat's milk because they have the same casein structure. So there's molecular mimicry there, right? If they react to melon, 92% of them will react to avocado fruits or watermelon because of that amino acid sequence. Okay? Now, what's important to know about this is you'll have sometimes patients get a lab test back, and when they get the lab test back, they'll like show up, they tested positive for scallops. And they go, that's impossible, I don't eat scallops, how could I make antibodies against it? If I see scallops, I want to vomit, I would never eat them. So this lab test is wrong, but it may not be wrong. It's just the fact that they're eating another food that is very similar to the amino acid sequence of scallops. So when they test their blood with that similar amino acid sequence, it tests positive. So this is the key concept here, misleading lab results due to cross-reactivity. If the immune reaction to a particular food protein shares a similar sequence of amino acids with another food protein, the patient may test positive on lab results, even if the patient is no longer consuming that food. This is due to protein sequence cross-reactivity. Okay? Does that make sense with everyone? So realize that certain food proteins are very similar. So when someone has antibodies to those food proteins, even though they're not eating other food proteins, they may still test positive because of that amino acid sequence similarity, or they call it homology, that they're the same. Okay? So food proteins interact with each other, and the way to really think about it is not to think of an immune reaction against a food protein, but think of an immune reaction against an amino acid sequence. And many foods have similar amino acid sequences. That's why people have sensitivity to all dairies, or all milk proteins, or all grains, or all fish products, because of that similarity in structure. Okay? Now, dietary proteins can cross-react with body tissue proteins, not just with each other. This is another key concept. So cross-reactivity can occur between dietary proteins and human tissue proteins due to molecular mimicry. So here you have two proteins. These two proteins have similar structures. And then what you see here is a circle, and the circle is the antigen-antibody binding mechanism. And if the proteins are similar enough to each other, like these two proteins here, or these three proteins are all different proteins, but they have the same structure so that when they fit in the key and keyhole, they hit the same amino acid sequences, and now they have molecular mimicry. That's the major concept. So they have found cross-reactivity. Here's a study with shrimp proteins. And if you take a look here, there's 100 proteins in shellfish. Of these 100 proteins, there's a sequence of amino acids, about 300 of them. And then the sequence of amino acids is right here. So you have shrimp and you have human. And human here is tropomyosin. Tropomyosin is a target protein for autoimmune ulcerative colitis, for example. Tropomyosin is also found in antibodies to people to their bladder. People that have interstitial cystitis sometimes have tropomyosin. So you can have someone who has antibodies to tropomyosin. They've lost tolerance. There's memory B cells responding and attacking tropomyosin. Now they eat shrimp. They make antibodies. That protein has the same structure. So those tropomyosin antibodies get made to shrimp and to tropomyosin, and boom, you get this molecular mimicry response. So they've looked at these in different studies, and they found that, shrimp tropomyosin binds to Ig antibodies, and patients that have this tropomyosin antibody, shrimp can actually be a trigger, and they found that it cross-reacts with human endogenous tropomyosins, and people that have ulcerative colitis, if they have antibodies to shrimp, they can have their autoimmune disease flared up from that specific food to cell tissue cross-reactivity. So cross-reactivity could be against foods to each other. It can be against foods to tissue proteins. Now, everyone that eats shrimp is not going to have this reaction. So first of all, let's say you eat shrimp, okay? In order for a cross-reactivity response to take place, number one, you have to have lost tolerance. And you have to have antibodies produced. Now you also have to have antibodies to tropomyosin. So if you have antibodies to tropomyosin, you've lost tolerance because of susceptibilities, of genetic susceptibility, environmental factors. Now you have tropomyosin autoimmunity. Now if you eat shrimp and make antibodies to shrimp, then that can be a cross-reactive response. But for people that don't have those other factors, they have good tolerance, they don't have tropomyosin antibodies, eating shrimp is not a factor. Now you could also have ulcerative colitis, but you don't make antibodies against shrimp. Then you shouldn't have a reaction. So cross-reactivity only takes place when you have the specific autoantibody target protein and a food with antibodies to that food together. And then that's the mechanism. Now there's another one called neuromyelitis optica. Neuromyelitis optica is a severe form of demyelinating disease that impacts the optic nerves. People lose vision, they get blind, hits their spinal cord, hits their entire brain. They basically get MS, multiple sclerosis type patterns, demyelination, but it's a different target protein than MS. It's not myelin. It's aquaporin 4. It's a target protein. So what happens in this autoimmune disease is antibodies are made to aquaporin 4, which is a protein receptor on the blood-brain barrier. And once they get that, they get this inflammatory reaction that occurs in their nervous system, and it really hits the optic tract pathways because there's lots of aquaporin 4 receptors. And one of the things you'll see with them is they lose their pupil response. So here's a patient with neuromyelitis optica, and what you're going to hear is, you're going to see the lights go on, and you're going to hear the clicking is lights turning on and off. And what should normally happen is when you turn the lights off, people should dilate, and when you turn the lights on, people should constrict. But when you have neuromyelitis optica, there's so much inflammation in the optic nerve that those things don't happen. This patient came to my office not knowing they had NMO. Like, I don't feel well, I'm not doing well. I'm like, okay, do an exam, there's no pupil response. What is going on? Then we see upper motor neuron signs, and I was like, okay, we gotta get you worked out. So it was a neuromyelitis optica. Now, this patient already is on a 100% gluten dairy-free diet. Because, you always jump to those two foods when they're the most inflammatory and reactive. So there are food proteins that may cross-react with aquaporin, NMO, aquaporin 4. And the ones that have the same similar protein sequences are soy, tomato, corn, and spinach. And here's the amino acid sequence. You see that? Human, aquaporin, soy. Because if you make antibodies against soy, you're going to bind to aquaporin because of molecular mimicry. So these are the target sites of the autoantibody reaction. And this was a paper that we published in the Journal of Autoimmune Disease, where we actually did this with the neuromyelitis optica patients. We looked at those food proteins, we looked at the amino acid sequences, and then here you can see these plot analysis. The blue are controls and the red are subjects with NMO, and we checked all the different neurological antibodies and all the food proteins. There was a statistically significant difference between these two. So people that have NMO not only had this risk, but many of them had this antibody reaction. So what do you think? How about this gluten dairy-free diet for people that have aquaporin autoimmunity? Right? That's the point I'm trying to make, is there's sometimes what's called cross-reactive reactions. They're very, very specific. It's not just gluten. Many foods you'll find on the autoimmune paleo diet may not be appropriate for some people with autoimmunity because of specific cross-reactivity. Now, let me explain something too. When someone eats a food protein and they have autoimmune disease, it may just cause general inflammation with them, or it can devastate them. When someone eats a food protein and it devastates them, it's probably cross-reactivity. Do you guys understand what I'm saying? Because the antibody molecular mimicry response is very aggressive. It's a very specific B cell binding that then causes the ultimate soldiers of the immune system to destroy it, NK cells and T cells. Do you guys understand? It's a big deal. So you always want to see, like, yeah, I had a salad and like I couldn't get out of it. I couldn't walk the next day at all. I couldn't see anymore. NMO, you're like, okay, well, maybe it was a cross-reactive reaction. Okay, and this is the real world of autoimmune disease. They have these different molecular mimicry mechanisms. This is a study that was published on cross-reactive rheumatoid factor in rheumatoid arthritis extracellular dysfunction, and they were looking at anti-DNA antibodies and in rheumatoid factor. And one of the things they found out is that rheumatoid factor, you guys measure rheumatoid factor? Rheumatoid factor cross-reacts with anti-DNA antibodies. So if their rheumatoid factor levels go up, they actually get cross-reactivity against their own DNA. This is an antigen cross-reactivity. And rheumatoid factor is just IgM and IgG agglutinated together. Look at this paper. Anti-TPO antibodies from patients with Hashimoto's encephalopathy bind to cerebellar astrocytes. TPO antibodies have molecular mimicry with the cerebellum. So TPO antibodies not only bind and attach to thyroid, but they also attach to the brain, specifically the cerebellum, which can cause ataxia and disequilibrium patterns and a whole host of cerebellar degenerative disease signs. And that's important to know, because what we're learning is that here's what's depressing. We don't have research for every autoimmune disease, for every antibody, and what it cross-reacts with. We'll get little glimpses of this and then try to understand the autoimmune disease and disease more. So I remember I always loved neurology. I just had a passion for neurology, and then I wrote a book on the thyroid, and I had all these thyroid patients starting to call me because they read the book and were interested in seeing me to help them with their case. And I'm doing my neurological exam, I'm going, oh my gosh, so many of them have neurological deficits. Like I would have never imagined that were to happen. And I remember when I saw this paper come out, I literally froze for like 2 minutes. Because I was searching for myelin antibodies and neurological antibodies, because when you find one, you find the others. And they didn't have it in a lot of cases. And then I go, okay, it doesn't even have to be there. Just the fact they have thyroid antibodies means they can actually have cerebellar inflammatory reactions. So they get termination tremors, they get dysdiadokinesia, they close their eyes, they're all over the place, and they're getting a neurodegenerative disease from an autoimmune mechanism from their TPO antibodies. Okay? So that's important to know. Now, here's another paper. It was the gut-joint axis cross-reactive food antibodies in rheumatoid arthritis. So they went through and checked food proteins that bind and cross-react with cartilage proteins, and what they found that was significant was gliadin. Gluten was significant, and casein. Gluten and casein were cross-reactive with cartilage antibodies. So people that have RA should definitely be on a gluten dairy-free diet. Most autoimmune disease patients have severe reactions to gluten and dairy, but here they found specific cross-reactivity between RA tissue antibody proteins and gluten and casein. Okay? So these are there. Now, paper that I was involved, I was the chief author, we published in the Journal of Thyroid Research. So I remember when I was working on my PhD, I was working with Dr. Risto Vijdani. He's a PhD immunologist, and most of you guys know him. He's the best. He's awesome. Most of the amazing papers in your notes are his. Just a phenomenal guy. He goes, what do you want to do? Well, what can I do? He goes, anything you want. I'm all, anything I want. He's like, yep. I go, okay, well, I have a lot of thyroid patients. Let's check cross-reactivity for foods that cross-react with the thyroid. He's like, all right. So we did. So, and we actually did publish it. So this was a paper that we did. Here we are, this is a lab. So you're taking these wells, you get purified proteins, you cross-react them. Then we're like, what parts of the thyroid should we cross-react with? So, by the way, this is the amino acid sequence of TPO. Okay, so you print out the sequence of the protein, you figure out what the target autoimmune sites are, then you call a lab that makes what are called monoclonal antibodies, which are high affinity, remember low affinity versus high affinity? High affinity pure antibodies for that target protein. So we had them make high affinity proteins for us for TSH, TPO, thyroglobulin, T4, T3, thyroid binding globulin, and 5-deiodinase. Why? Because he said anything you want. So here we go. Because that's everything I wanted. So we contracted an immunology lab. They made us, we got purified monoclonal antibodies, high affinity, and we took 200 foods. We took 200 foods and we had to purify them through dialysis, so they're nothing but pure protein. So then you take pure protein and a high affinity monoclonal antibody. You put the pure protein in a well, stick it to a lyso, so it's attached there. Take a monoclonal antibody with high affinity only for that target protein. You mix it together and you see if they cross-react. And that's what we did. We did it for the thyroid. And we did it for some other conditions too. But, and this is what we found. Of the 204 foods we tested, we didn't find any cross-reactivity with TSH. Look how many we found. We found about 40 cross-reactivities with T3 and with T4, and about 18 strong reactions with T4. These are some selected foods with cross-reactivity. Look at gelatin, scallops, almond. So these are cross-reactive interactions. So what does that mean? That the person has to have lost their tolerance, and not only that, they have to have Hashimoto's, but actually make antibodies to scallops. But if they make antibodies to scallops, well, scallops can bind to their T4 and T3. So they're taking T3 and T4, but as soon as they eat scallops, those antibodies attack their T4 and T3, and it neutralizes their hormones, and now they have no effect from it. You understand? That's the concept of cross-reactivity. So how appropriate are these nutrient-rich, organic, gluten-free, dairy-free choices for Hashimoto's patients? Here it is being prepped. Well, tuna cooked and raw, cross-reacted. So there goes that. It's on the autoimmune paleo. Who cares? Cashew cross-reacted with T3, T4 using latex gloves. Found latex having a protein that's found in avocado and in latex gloves cross-reacts if they're preparing their food and make antibodies against those, then you can have cross-reactivity. So that's called, these are all food proteins that directly cross-react with tissues. Does that make sense with everyone? Okay. So one of the things that we're going to talk about is when we do food dietary management. Like right now, because of this research, I can take a Hashimoto's patient and go, okay, I can test you for your food proteins and find out which ones have molecular mimicry directly with your thyroid axis, because we've discovered those and we published them. So now my dietary regimen is much more precise for a Hashimoto's patient than it would be for another disease where I don't know what all the cross-reactive foods are. You guys with me? We did the same type of research with type 1 diabetes. And he goes, well, he goes like, what do you want to do next? I'm all type 1 diabetes. He goes, why? He goes, well, my wife had a sister with type 1 diabetes, and she said when she was a kid, she was going to marry someone that would cure autoimmune diabetes. I know, isn't that great? So I can't cure it, but we went and studied type 1 diabetes. We found all the cross-reactive foods for type 1 diabetes. We published that in Journal of Diabetes Research. So the goal is for us to take different autoimmune diseases and start purifying foods and doing it. But I got to tell you, it's labor intensive. It's very, very expensive. There's not funding for it. And like, we're not going to be able to do it, all of them. So right now we have 2. Maybe we can do RA next or something, but it's a lot of work. Okay. And also, a lot of journals don't care. A lot of immunology journals are like, yeah, that's cool, that's great, but who's going to ever use this? Like literally, that's what you'll have some reviewers say. Submit it to a journal, yep, science looks good, data looks good, analysis looks good, methodology looks good, but it's not relevant to clinical practice. It is for us. So anyways, so it takes time. Now, when you look at food reactions, so you have, what we talked about so far is specific cross-reactive reactions, and then you can have diverse immune reactions. Diverse immune reactions means you just trigger the inflammatory response, okay? So we spend a lot of time talking about specific cross-reactive reactions. Let's talk about gluten for a sec, because the gluten does both, and I can move on. I mean, everyone already knows gluten is bad for inflammatory conditions, autoimmunity, and it's not because the protein has been genetically modified. Part of the reason why gluten is so reactive now is because it's been hybridized. So hybridized isn't genetically modified. They just keep putting in different seed combinations together. And what they found is about 5% of gluten proteins are new proteins. So you can hybridize new proteins. If you combine them together, you get different immune reactions. So this is now a new antigen. And in the immunology literature, they use the term modern wheat versus native wheat. And modern wheat is a different protein. Okay? So if most of you, when you were young, you were eating native wheat. This in the past 20, 30 years, now you're eating modern wheat. So you're like, whoa, when I was a kid, I ate Wonder Bread and all this stuff. I didn't have any reactions if you're gluten sensitive, and now you do if you're gluten sensitive. It's a different protein. Okay? Some countries still use native wheat, like France. So some people don't have reactions in those countries. They only react to the new proteins. Some people have reactions to both. Now, when you look at gluten sensitivity, remember that there's a difference between celiac disease, we talked about this in the GI course, and you have the gluten exposure with the gene, specifically HLA-DK2 and 8 genotype, and then what that genotype does, it makes T cells significantly react. That's the feature of celiac disease versus gluten sensitivity. And once they have this genotype 2 and 8 with the transglutaminase 2 antibody mixed together, those genetic variations, their exposure to gluten causes severe intestinal destruction because they have an overactive T cell response. That's celiac disease. Gluten sensitivity is just an immune inflammatory reaction to gluten. And celiac disease is this profound T cell response based on their HLA genotype. Gluten sensitivity, there's many different types of genotypes, HLA DQ 1, 4, and various other ones, and some we don't even know yet. And they can cause inflammatory reactions to tissues as well. Now, if they have transglutaminase 2, the target protein is intestinal lining. Transglutaminase 3 is skin, and transglutaminase 6 is cerebellum. So there are some people that just have antibodies to transglutaminase 6, which means their inflammatory response will be against their brain. Some will have it just against their skin, okay? Now, in the literature, there's many people, many immunologists saying it doesn't have to be celiac disease. Non-celiac gluten sensitivity is significant. It is serious. People have significant inflammatory reactions with it. And basically, in this paper that was published, there's an international consensus that, yeah, it is a problem. Then in people that don't have to have celiac disease, just gluten withdrawal is a key issue. And you have to consider gluten removal with people that have autoimmunity and inflammation. And remember, cross-reactivity with gliadin takes place with cerebellum that we know about. This is a study with a patient that has cerebellar and gluten sensitivity. You can see the demyelination, and within 15 months, you can see the progression. It can be very significant for some people. And again, whenever you see serious pathology develop from a food protein, you probably are seeing cross-reactivity. And this is a great example of it. You see demyelination and MRI change significantly. That's not just inflammatory reaction, that's cross-reactivity. Okay? And there's been numerous papers published, some on synapsin with gluten, which is another target protein, multiple tissue proteins, pancreatic islet cells with gluten, and the brain and the thyroid, as we showed. So just realize that gluten is a major issue with people that have autoimmunity. It's very inflammatory. They're new wheat proteins, they're new antigens. They have a lot of molecular mimicry. The amino acid sequence of gluten is very similar to many tissue proteins in the body. So there's probably many unreported cross-reactivities with gluten we haven't even learned about yet that take place. And unrelated to whether they have intestinal destruction or without intestinal destruction, which they call enteropathy. Autoimmunity can be flared up with either celiac disease or gluten sensitivity. Celiac disease is just going to be much, much more aggressive. And when you check for gluten sensitivity, make sure you realize that there's different peptides of gluten. So this was a study in gastroenterology where they just looked at alpha gliadin. By the way, if you're looking at lab test, if you just see gliadin, it's alpha gliadin. So if someone gets screened for alpha gliadin, you're going to miss the other branches of gluten. Just think of gluten as a protein. Remember the protein structure with all the different amino acid sequences? Think of each of the amino acid sequences as branches. So there's alpha gliadin, gamma gliadin, and there's different types of gluten MERs, 17 MER, 5 MER, 21 MER. There's different peptides. Like in this study, these are all the patients. You had 1, 2, 3, 4 of them test for alpha gliadin. The other ones tested for these other reactions. So when Cyrex, Dr. Vijnani, was making a gluten profile to test for gluten sensitivity, he's like, I'm not checking alpha gliadin only. I'm going to miss it if I don't test the entire branches of the gluten proteome. So this is what this test will do. It'll check for all the different reactions to gliadin. Make sense? Because if there's one protein more than another that's going to cause inflammation for autoimmune disease, it's going to be gliadin. A lot of people will have alpha gliadin done even at their hospital or their GI, because a gastroenterologist will do it, but it'll just be alpha gliadin. So they may think they're okay, but you want to check the whole protein. The whole protein hasn't been checked and they have autoimmunity. You want to make sure you only rule out gluten sensitivity when you have this test clear. This is Cyrex Array 3X. And if you take a look at it, there's a couple of key features here. See how it says wheat in the very top? Let me explain what this is. When you do antibody testing, if you take entire wheat protein and purify it, you get a lot of different cluster of amino acids in there, okay? So when you have an antigen-antibody reaction, because of other mixture of proteins in there, you may not see the reaction because there's too much other stuff in the way. So then what they do in the lab is they just take out, instead of the entire wheat proteome, they'll take out branches of the wheat. You see what this is? Like here, wheat agglutinin, that's a lectin portion of wheat. Then here is alpha gliadin, gamma gliadin, omega gliadin. These are all different branches. And they isolate only that branch in a well for the antibody response. So that increases the chance you'll find the signal. So you may have someone not show up with wheat at all, but they only show up with gamma gliadin. It means you found the immune reactivity section of the wheat protein. Any one of these showing up positive is a gluten sensitivity. Okay? So this is really the most thorough way to check for these reactions. And then if you look down here, transglutaminases, these are autoantibodies for self-tissue. These aren't antibodies against wheat. Transglutaminase is a self-tissue antibody. If you see transglutaminase 2 positive, that is intestinal autoimmunity. Transglutaminase 3 is skin autoimmunity, and transglutaminase 6 is brain autoimmunity. So the top port checks wheat, the whole thing together, the whole protein together, and then it clears the debris and checks the different branches of the wheat. And then it checks on the bottom the autoimmune reaction that can take place with it. So this is the very thorough profile. And if people get exposed to gluten, we know we can take flavonoids to block the response. These are the known ones. We talked about this in the GI module. I just want to list it again here. And the other key issues with gluten is that they can cross-react. So gluten has amino acid similarity with dairy and other grains. So when Dr. Vijdani was organizing Cyrex panels, array 4, he goes, I want to check all the other food proteins that have cross-reactivity because of their sequences with gluten, which were these grains and these milk proteins. So that became array 4. So one of the things you always want to check with people that have gluten sensitivity or celiac, history of it, is to see if they have cross-reactivity with these other grains. Now remember, with that slide I showed you with the epidemiology of autoimmune diseases, and the concordance rate with twins with celiac disease was like 87%. It was the highest of any other autoimmune disease, meaning you're going to see patients that come in and say, I have sensitivity to gluten. You go, yes, okay, great. And you have celiac. Let's say they have the HLA-DQ2 in their family. That amino acid of gluten is also similar to milk and similar to other grains. And I think this is why you've seen this explosion in trend in paleo, autoimmune paleo, because so many patients feel better, not on a gluten dairy-free diet, but on a completely grain-free diet. And that's because of, most likely, molecular mimicry. Those amino acid sequences are very similar to each other. So this is just a slide that shows you like intestinal permeability, gluten reactivity. These are the cross-reactive foods. Always check those. And whenever you have a person that's gluten sensitivity, not only should you check the cross-reactive foods, you should also see whether they have the autoimmunity responses to gluten, which is transglutaminase 2, intestinal, 3 skin, 6 brain. And then gluten has been shown to cross-react specifically with GAD65. This is causing type 1 diabetes and cerebellar ataxia, cerebellum, and islet cells. So good research on gluten causing cross-reactivity with type 1 diabetes target proteins in the brain. So these are also things to consider. Okay? So gluten causes both cross-reactive and diverse reactions. So everyone understand cross-reactivity now? Like the concepts and so forth? Okay. We're going to talk about it more when we get to treatment applications. I just want to go over food concepts. Now let's talk about other dietary triggers in autoimmunity. The major breakthrough recently was salt. Salt is a huge trigger for autoimmunity. Sodium chloride drives autoimmune disease by the induction of pathogenic Th17 cells. So let me show you what happened. Let me tell you what happened with this. They did this, there were some laboratory researchers studying different T cell proliferations, and they found one of the dishes had really, really, really high production of Th17. And it was just like a group of dishes there. Then they go, well, how did that happen? So then they looked at all the researchers, and they found the new researcher on the team, which is like a new grad student, put too much salt in the dish. This is an in vitro test, and the Th17 cells proliferated. And they go, okay, well, that's interesting. It's an in vitro response. Since then, they've done animal and human studies with sodium intake, and they find it actually activates Th17. So what does Th17 do? Th17 adds fuel to the fire wherever the autoimmune disease is. So many papers have now shown that salt activates the T helper 17 pathway. okay? And I'll just show you some of these abstracts here and get to this illustration here. And then they found a specific protein and immune mechanism that activates the Th17 pathway. You can see it here. Sodium chloride activating this P38 protein here and triggering this release of Th17, which then flares up the autoimmune disease response. And then they also found CFOS changes here that activate the inflammatory IL-1 pathway for autoimmune disease. And they've done some observational epidemiological studies of patients with multiple sclerosis, and they demonstrate association between excessive salt intake and higher number of flare-ups. And they found excessive salt intake associated with higher risk of developing RA, also when combined with smoking. And when people have a high sodium diet, they can have their inflammatory response be impacted from it. And there's multiple review papers now on salt and things like MS, RA, lupus with salt intake. Okay? So they then go, well, what if we gave them potassium? What if we took patients and salt's an issue, but potassium antagonizes it? What if we do a study with autoimmune patients and we give them potassium and see if that has an impact? So they did the study. 49 healthy participants received low salt, high salt, followed by a high salt diet plus potassium supplementation for 7 days respectively. In the participants, IL-17 levels in plasma and peripheral blood mononuclear cells were significantly increased after a high salt diet, which is dramatically reversed with potassium when potassium was supplemented. Wow. Wow. Yes. I saw this and go, okay, new tool. Got a new tool in my bag. So, if I have patients who have autoimmunity, I will make sure they take potassium bicarbonate. I like bicarbonate the best. It has the most pH modulating effect. And if they have a salty meal, have them load up on some potassium. And I like to use about 2,000 milligrams. So here's the clinical rule. Patient with autoimmunity should consume potassium if eating salty foods. Now, there are a group of autoimmune patients that have very low salt sodium, like remember the adrenal autoimmune patients? They have aldosterone. They can't retain salt. Those people need to consume salt. It doesn't flare up their autoimmunity. They actually only function and get the blood pressure up when they consume salt. So remember, adrenal autoimmune patients, they don't make enough aldosterone. They can't retain salt. Salt's not an issue with them. But for MS, lupus, other autoimmune diseases, they found salt as a key factor. So one of the things you're always going to have to do with your patients is they go, I just had a flare up. You're going to have to work backwards and go, what happened? And you might be looking at food proteins, but don't forget salt is now a factor, okay? So you want to, if you see that association take place with your patients, you want to, let them know that they're probably very sensitive to salt as a trigger. And in that case, if they do have anything salty, always take some potassium with them, okay? Now, potassium capsules are limited in size to, 100 or 200 milligrams, but they usually need about 1,000, 2,000 milligrams to have an effect. So you can use a potassium bicarbonate powder. That's what I use, the scoop. But if they travel, they're just going to have to take enough capsules to get to 1,000 or 2,000 milligrams. They're not going to overdose with 2,000 milligrams of potassium. But it's a key thing with autoimmunity. They may have to even play with the dose of potassium to find the best dose. Make sense? What a great study that was done, right? Now we have an application that we can use. So salt, it's there. Know it's one of the triggers. Teach your patients about that being a trigger, and the way you can deal with that is potassium. Okay. Now, food coloring and autoimmunity. So, food coloring is a major issue, and food coloring is associated with neuroinflammatory autoimmune reactions and so forth. But the basic concept is, here's the key concept: antigenicity of an individual food protein may increase with food coloring due to an inability to properly digest, leading to increased peptide antigen presentation to the intestinal mucosa. Foods with food coloring should be avoided by those who have impaired oral tolerance. This is a diagram from a paper Dr. Vijnani published, and it shows you different food colorings to proteins. You see this illustration here? The scissors represent enzymes, and they're breaking down the amino acid sequence. Okay, so remember, the antibody can only bind to an amino acid sequence, right? If the amino acid sequence is broken down to individual amino acids, there's nowhere for the antibody to bind, hence no immune reaction. This is why it's so critical to have healthy breakdown of proteins. So when people have digestive enzyme issues and they can't break down their proteins like hypochlorhydria, that protein stays as a target for antibodies. Okay? Also, if a patient had a food they ate and had a significant reaction, you should immediately have them load up on digestive enzymes, especially hydrochloric acid, to break that protein down as soon as possible so it doesn't become a target marker for antibodies. What food coloring does, which you see represented in yellow, is it prevents enzymes from breaking down the protein. So they keep maintaining that amino acid sequence, which means antibodies can still bind to it. Okay? And in this study, it goes on to show when they take a food protein, and the blue represents a normal food protein, and the red is with food coloring, you can look at the time to minutes on the x-axis, and the y-axis is digestion. So you can see as time goes on in the blue, they start to digest their foods. When they take the same food protein, add food coloring to it, it doesn't break down as efficiently. If it's not breaking down, it's a target site for antibodies and inflammatory triggers. So with patients that have autoimmunity, they may get exposed to a food that has food coloring and they can't break it down, and then they have a significant reaction. Like jelly beans, gelatin, gluten with food coloring. I only had one jelly bean and now I'm in bed for 3 days. Yeah. Don't eat a jelly bean. Okay. Don't add food coloring. Don't have gluten beer St. Patrick's Day with green dye in it. Not the best idea. Have the regular beer and then not be as bad, but don't do it at all. The food coloring is a problem. So this is just an illustration here, which you can see that concept, the undigested food protein. On the left-hand side is normal, healthy digestion. The food protein is broken down to individual amino acids, no immune reaction. So undigested protein is broken down to peptide, amino acid, and then intact mucosa, everything goes through. What you don't want is lack of digestive enzymes or food coloring. Food protein is this amino, it's not broken down. You still have peptides. These peptides get through, especially if you have permeability, and that causes ongoing immune triggers, inflammatory responses as well. Okay? So food coloring is an issue. Salt is an issue. Okay? Here's another one. Blood sugar control. So blood sugar control means spikes of insulin, spikes of glucose. Fluctuating blood sugar levels trigger autoimmunity. So you can see here a study where they looked at excess glucose activation of the immune system. So when glucose levels become elevated, they directly activate toll-like receptors. Toll-like receptors are what's going to trigger the inflammatory NF-kappa B response. It's going to put fire on autoimmunity. Okay? And here you can see another diagram. Overnutrition, hyperglycemia, directly activates NF-kappa B, activates INOS, activates the nitric oxide pathways associated with autoimmune flare-ups. Interestingly enough, hypoglycemia has also immune triggering events. Your biggest clue that a patient has blood sugar issues is they have changes of energy after they eat. They feel tired. That means they're not getting glucose in their cells. They're getting insulin surges. That glucose is getting converted to fat, and they get exhausted because the conversion of glucose to triglycerides requires energy. If they're hypoglycemic and they eat, they feel energized. The normal response is no change in energy after they eat. Because I've had so many patients referred to me from other practitioners. I sit down and go, let's stabilize your blood sugar. We stabilize their blood sugar, and their autoimmunity goes into remission. These are the ones that have already eliminated every food. They're already taking more supplements than I've even heard of. And I go, we got to stabilize your blood sugar. Let's do that the first 3 weeks. We do that, and I've never felt better. Okay? So when you look at dietary considerations with autoimmunity, blood sugar stability is one. So I tell my autoimmune hypoglycemia, let's say I have a patient that has, let's say autoimmunity, let's say their autoimmunity is thyroid, and they are hypoglycemic. I say every time you get shaky, lightheaded, and irritable, say bye-bye thyroid tissue. Or if they're insulin resistant and have demyelination, I go every time you get an insulin surge and fatigue, say bye-bye myelin, because they're activating the response. So you have to stabilize blood sugar levels in addition to dietary removal of restrictions, that the inflammatory response is so aggressive with dysglycemic spikes. Okay? So you got to get them to the point where eating meals doesn't cause them to feel energized, which is a sign that their blood sugar was low, and eating meals doesn't cause them to get tired. It's a sign of insulin resistance. Okay, those are the key things. Now, when you look at dietary proteins and autoimmunity, nightshades are part of the problem too. We know nightshades can be a major trigger for many patients with autoimmunity. Nightshades are tomatoes, eggplants, and peppers. So here's a list of nightshades: white potatoes, not sweet potatoes, eggplant, tomatoes, bell peppers, peppers but not black pepper, paprika, red pepper flakes, cayenne pepper, for example. And there's two key things with nightshades. One is they have alkaloid properties. And these proteins, these alkaloid proteins can flare up autoimmunity in some susceptible genotypes. And then lectin is the sticky portion of the plant protein. For many people, they have significant reactions when they eat nightshades. So here we see susceptible genotype. Alkaloids bind to immune cell receptors and promote inflammation, promote agglutination, development of altered proteins. You guys know what agglutination is? Let me explain this. You have a protein. Proteins stick together. That's agglutination. What happens when you take proteins and stick them together? You make a new protein. What happens when you make a new protein? You get an immune response as a neoantigen. Some people are very susceptible to agglutination. People that eat nightshades have agglutination. And are sensitive to them. So they get this agglutination response with these foods that have high lectin properties. For whatever reason, genetically, they stick together more often with them. So they eat high lectin foods, and they're prone to agglutination. Those food proteins become new peptides, and they create an inflammatory response. Now, there's the alkaloid portion of the food, which is unrelated to agglutination. So what that means to you is they may not show up on a food sensitivity test because of the alkaloid immune response unrelated to the lectin. So here's the clinical parole. Some patients with immune sensitivity to nightshades have genetic susceptibility to alkaloid binding mechanisms leading to inflammation, but is not associated with agglutination. Therefore, they will not show elevated antibodies, but notice a difference when they eliminate them from their diet. Be aware of that. Some people, you're not going to find nightshades on the food test. They'll have to actually remove it because of the alkaloid properties that are immune reactive, not because of the lectin. So you might want to do with an autoimmune disease patient like a trial of just going nightshade free for a while and seeing how they do. Now, lectins are sticky portions of foods, and nuts and seeds have lectin properties. And this is a paper that Dr. Vijani published with Jema Lambert. Jema's here? You guys, Jema, it's your paper. She published this paper. You can talk to her, expert in lectin foods. And they went and showed significant reactions with lectins in different autoimmune conditions. And we know that lectins have a significant impact on autoimmune reactivities. And lectins in beans and peppers and nuts, these foods can be an issue with many, many people. And just to kind of summarize the key concept, here it is. This is agglutination. Agglutination is sticking of compounds together. Some people are very sensitive to agglutination. Now, your biggest clue you have to be worried about agglutination is if you ever have a patient come in with high rheumatoid factor. Anytime a patient has positive rheumatoid factor, suspect exaggerated agglutination responses genetically. Carefully screening for sensitivities to lectin foods with antibody test and an elimination preference diet challenge. So when you look at rheumatoid factor, it's agglutination of IgG and IgM together. That's what rheumatoid factor is. So they have the susceptibility for agglutinating, which makes them very likely that they will have sensitivity to foods that are high in lectins. Some autoimmune disease patients, but not all, have significant inflammatory reactions with lectin compounds. I know now there's a trend with everyone being lectin-free. You guys, not everyone needs to be lectin-free. I can tell you that for sure with autoimmunity. But there are some that will have a night and day difference when they go lectin-free. Okay? So these are the nuts and so some people that have to go beyond just gluten dairy-free, they have to avoid lectins altogether because they have the susceptibility for these compounds to combine together. And then we also know there's some research that shows if you heat your food and you can change the lectin content of them. So some notes in there about how some research has been done to inactivating lectins from heat digestion, which can make some people tolerate some degree of lectins if they have some mild sensitivity to it. Some people can't handle them if they're cooked or raw. It's just they're not getting enough of the lectin breakdown to make them inactive. So be aware of that as well. There are people that will go, hey, no, if I inactivate lectins through heating, I can eat them. Some cannot, because you don't get complete inactivation. Despite this paper showing that you may, there's just clinically you won't see that. Okay? So those are the key things with foods, specific and direct immune reactivations. Now, at the end of the day, as a clinician, you're going to go, well, what do I do? What do I do? Can't eat anything? Can't even eat salt now? And the answer is yes. I know it sucks, but yeah, maybe. Okay? Because here's the thing. First of all, let's talk about dietary considerations first, okay? Let's talk about what the choices are. You have, in this scenario here, dietary sodium and stabilizing blood sugar. So that is a must for most people. You're going to really have to look at their salt intake. If they're going to eat salt and have autoimmunity, make sure to take potassium with it. Figure out how that impacts them. For some people, it is going to be very obviously a factor. For others, they're not going to notice much with it at all. Okay? The ones that really have a direct reaction to it, you should probably have them avoid salty foods as much as they can. And if they do eat something salty, make sure they have potassium. Okay? And then you have the choice of going dietary protein restriction. So do you go gluten-free? Do you go dairy-free? Do you go cross-reactive grains? Do you go nightshade-free? Do you go lectin-free? And ultimately, you're left with these kind of diets: gluten-free diet, paleo diet, and autoimmune paleo diet. Gluten-free diet is pretty much just gluten and usually dairy. Paleo diet is getting rid of grains. Autoimmune paleo diet is getting rid of grains, lectins, and nightshades. So when most people are doing a general diet, they're seeing the best effects, many of them, with the autoimmune paleo diet. And that's been quite popular. Now, there's some ridiculous stuff going on, I don't know, in forums that I'm hearing about, that people are going, if you have autoimmunity, you only need to be on autoimmune paleo diet for like 30 days and then you can switch. Like, no, you can't. Where did someone even come up with that? All of a sudden, like, your immune system is like, nope, that's all I needed was 30 days. I know it sucks after 30 days. So for your psychology, you can now eat it. It's not how it works. Some people will get sick. Now, I have a little chart here. This is basically your gluten dairy-free diet, and then this comprehensive permeability diet sheet is the autoimmune paleo diet, okay? So you have to decide what you want to do. Now, patients are going to go, how long? Here's the answer: as long as you have an autoimmune disease. To what degree can vary. Now, you have to understand, the basic level is to get patients gluten-free. Now, in a real world, your ability to change your diet is going to basically be based on the patient's motivation to change your diet. Like, if they don't care, they don't care. If they aren't willing to try, they're not willing to try. You're going to see a whole spectrum of patients. You're going to see patients coming in that are on autoimmune paleo and they're only eating 4 things left. And that's actually really bad because it makes them lose their gut diversity. They have other problems with it. Then you have some patients that will fight you, I don't want to avoid gluten. What if I just eat occasionally? And that's your own conversations you got to have with your own patients in your own practice. I can tell you, your tone does make a difference. Like, if you look at someone and go, I think maybe, like, if you want to, you should really try to avoid gluten. That doesn't have the same response as, like, you need to stop eating that. You have neurological autoimmunity, and it's going to flare it up because it cross-reacts. I've seen you 5 years from now, and it's not good. Totally different response. Does that make sense? Okay, don't be a wuss. Look him in the eye and tell him what you believe. And if you guys are seeing transglutaminase 2, like really positive in a lab test, and you're seeing myelin antibodies, again, unless you're serious enough with them, they're not going to get it. If you see rheumatoid factor, you see agglutination, and they react to lectins, it's a big deal. Now, ultimately, a patient's going to do what they want to do. And for the most part, most of us practice functional medicine. Most of us are in private practice, not all of us, but patients that we see are highly motivated. So we usually get them to do. They just need direction. So when they need direction, it's great, because you're not going to convince someone to change their lifestyle if they're not going to change it. My response is, you got to see me when you're ready. I'm here. What that usually means is you need to get sick enough. When you're sick enough, come back. Have a nice day. Here's another good clinical pearl for you guys. This is the practice management pearl. Let patients go. You want to energize your practice and your motivation and be inspired to practice every day? Release patients. Bye-bye. I can't do anything for you. Like, there's nothing left for me to do because this is an ongoing trigger. I don't want you to appear every day mad, you're not getting better, and other things, and you're not, I can't, there's nothing for me to do. I don't have another supplement to give you. I don't have another diet to change. I don't have something easier for you. But I'm here if you need help. And right now, these are the key factors. I can't do it. And sometimes it's not diet. Sometimes it's like they won't change their job, or they won't change their environment they're in. And it's obviously a trigger for them. So you can't do magic. You're only a clinician. All you can do is guide them. And if you see a hurdle where they can't get in past it because it's a factor, because it's unhealthy for you to work with them. It's unhealthy for them to be in your office. It's unhealthy for them as well. Does that make sense? So, there's a couple things you have to understand with autoimmunity. You have to understand the landscape so you know what's realistic and not realistic for outcomes. And you got to know you got to work through it, and every patient's going to be different. That has to happen so you have the psychology to continue to see autoimmune patients and not want to quit. The second part is there's some patients that aren't ready to go to the level they need to get better. If they're not, hey, I brought my husband and he's got type 1 diabetes, he doesn't believe in this whole dairy gluten thing, then get out. Because a big part of my practice is dietary modification. And if you're not going to do dietary modification, I am not for you. You may want to look at the corticosteroid section, or you guys understand what I'm saying? This model, you can't push this model on people. All you can do as a clinician is educate, And I can tell you, every time I teach topics, I always get the question at the break, what do you do with a patient who won't follow your recommendations? I don't know. I'm not a psychologist. Okay. So anyways, there's gluten-free, there's dairy-free, there's autoimmune paleo. If a patient doesn't know and they want to be very, very aggressive, then they should do autoimmune paleo. The other key thing here is support oral tolerance. We're going to talk about that more when we cut to treatment. But then just to make sure they have a diversity of lots of food proteins. Because a lot of times when people go to autoimmune paleo, what that translates to them is, I have to stop everything I like to eat. I have to eliminate everything. Food is no longer a joy for me. I'm just going to eliminate because I'm sick. And then just eliminate foods. And they find, I only like these vegetables. I only like these foods. And they have the same diet every day. And then that changes their immune tolerance and their gut microbiome. And they get more autoimmune expressions. So you got to make sure if they're going autoimmune paleo and restricting that they're still diverse. That's a key factor. So, and then if you have the ability to look at cross-reactive foods, that does make a difference to see what kind of change that they can make with them. Okay? So you guys, those are the key things with foods. Gluten, dairy, grains, lectins, nightshades, blood sugar stability, salt, food coloring. That's what we at least have some great understanding of these. Now, what the X factor is there's some cross-reactive foods for certain autoimmune diseases, and we don't know what they are. Who would have ever thought shrimp protein with ulcerative colitis? I don't know. I would have never thought that. Would have thought scallops with T4? I don't know. So you got to do the best you can. Okay. Now, the next thing I want to cover is laboratory testing for food proteins. I got a lot of people upset with me at the last seminar. Some of you know why. Okay. So there's two types of food protein testing: antibody testing and cytotoxic testing. So let me explain these two. So cytotoxic testing is not an antibody test. And the main types of cytotoxic testing out there are MRT, mediated release testing, ALCAT, and LRA. So these basically measure non-specific cell size change to an antigen-presenting cell. And what you guys need to know about that is it's banned in most states, not even allowed to do it. Different results from the same patient, poor reproducibility, and high number of false positives. And with these cytotoxic testing, what they're basically doing is they're taking an antigen and seeing if the antigen-presenting cell gets larger. It has no specificity, has no sensitivity, and it varies all the time. And basically what they do is they take a well, they expose it to a protein, got bigger, positive, don't eat that food. That's cytotoxic testing. Here's some studies on cytotoxic testing. The reproducibility of cytotoxic food testing was studied with Alcott's same blood samples done in duplicate as well as reproducible retesting of the patient. And basically, they found that it was unreliable for the diagnosis of food sensitivity. They did the same thing with this study here, controlled study of food cytotoxic testing. Repetitive testing was performed, no reproducibility. This is a letter from one of the states saying it's completely banned because it's not an appropriate test. Here's the FDA showing that cytotoxic testing has no validity. Here's the Commission for Dietetic Registrations, the RA community showing that it has no credibility. Because it has no credibility, it's not a valid test. Just because a lab offers a test doesn't mean that it's a valid test. Okay? So I would really encourage you to stay away from cytotoxic testing. You won't find any good papers on it. And we had a discussion about this after the GI course because someone asked, what do you think about cytotoxic testing? And I said something I really shouldn't. And I said, well, if you do it, the only thing it tells you is that you don't know what you're doing. It's just something stupid like that. I was tired. It was Sunday. I had 2 days of lecturing. You can't gate things all the time, you know? It was a dumb response, but made a lot of people upset that we're doing cytotoxic testing. But then it was followed by, well, show me the evidence. Okay, well, there's lots of evidence. That it's not reproducible, that it's not effective, it's not the gold standard, and that's a fact. And then we go, hey, call all the labs you want. Post the studies that shows it works. You know why I said that? Because there's 0 studies. What some of these labs do is go, we patented this test, which is an application fee. Doesn't mean it's valid. We patented this test. Well, who cares? Publish it. Show that it's reproducible and reliable. Specificity and sensitivity are critical things in laboratory sciences. You have to know what they are. And if they're valid and they're the best, that's called the gold standard. The gold standard for foods is not cytotoxic testing. It is antibody testing. And trust me when I tell you, because I've been called by many state boards for people that are in defenses or different things, an expert, you can't defend it in a court. This patient saw this person, they did cytotoxic testing for this autoimmunity or whatever, and he's like, well, that's a shame. You can't defend that. If anything, it makes the practitioner look worse. Because doing a non-validated test that's banned in many states, that the FDA says has no reproducibility, and American College of Pathologists doesn't certify, CLIA doesn't certify, why would you ever do that? So that's a problem with cytotoxic testing. So be very aware of those types of responses. And then you have antibody testing. And then when you look at antibody testing, antibodies are very specific, high affinity. The most specificity you can get with foods is an antibody. That's the whole concept of adaptive immunity, very specific to that antigen. And the key with antibody testing, there's 2 types. There's ELISA methodology and there's microarray. And the gold standard is also microarray. The gold standard is ELISA, it's not microarray. So ELISA testing is the method of choice, okay? Now, here's a paper where they looked at microarray compared to ELISA. And they looked at correlation. So you guys are familiar with correlation, R values? R values, like a 1 would be a perfect correlation, that if this is positive, that's positive. And they did this with different food proteins. So they had one correlation, which was 0.8. And by the way, the percentage of that would be squared. So 8 would be like 64%. 64% correlation with an R value of 8 compared to ELISA. That's not good for microarray. This one was, I think it's a 81%. Well, people cite there's a 93% correlation. No, there was a 0.93 R value, which is 80%, correlation, or 81% correlation. And if you look at some of these other ones, look, the R value is 67, so 36%. And they vary from one food protein to the next. So some are down to 30% correlation, some up to 80, maybe 90, some are down to 60, some to 40. It's all over the place. So that's the key issue with doing microarray analysis for food antibodies. It's not the gold standard. Microarray is fantastic for cancer protein early detection. It's not good for food protein. Okay? So then here's what you see from these labs that do microarray. Here's one, by the way. One of the papers, Vibrant Sciences, LLC. Oh, that's not conflict of interest. And what they did is they go, oh, look, we have the study that we did, and we found a 97 to 99% accuracy doing microarray. What they fail to tell you when you actually read the paper is that they actually did a principal component analysis. They built an algorithm. Guys, if you build an algorithm, you can get 9900%. It's not the test. It's like this test plus this, plus that, plus this, plus this, plus this. There's a calculation that equals almost 100%. When you guys go on YouTube and you have the same stuff play over and over again, they did an algorithm on you. They know what you like. When you get ads on Facebook or whatever, they did an algorithm on you based on your likes to figure out what you want. It can be highly predictive. These papers that they're handing out to people to show that the microarray testing is valid are not comparative tests to ELISA. They are algorithms that are developed. So again, if you read these papers, look at the methodology here. It's pretty complex. If you don't understand predictive study analysis and regression and primary component analysis, which I had to learn during my work, research work, it's hard to understand. So you just think, oh, these results are actually really accurate. Microarray is very specific, but they're not doing microarray. They're doing predictive models. And then they're brushing off these papers as if they're doing microarray. This is unethical. Same thing happened, look at this one. Determination of B-cell epitopes in patients with celiac disease. Peptide microarray. Boom, boom, interesting. Vibrant Sciences, LLC. Oh, here's the result. Oh my gosh, you did machine learning algorithms. That's not the same thing. This is what upsets me a lot. So then they have people trying to diagnose, manage autoimmune disease with a test that's not gold standard, and the practitioner is being lied to, and they think that it's gold standard. And the studies they're referencing are more algorithms, predictive models, not actual microarray. Do you guys see that? Okay. So that's why we don't do microarray, because it's not the gold standard. And be careful of the papers that you get and actually read them. Okay, so the gold standard is ELISA testing. Now, I work with Cyrex. I prefer Cyrex. And part of the reason I prefer Cyrex is because they check for cooked and raw, which is really important. But they also check for lectins. You guys remember we talked about lectins being sticking together? So remember, like when you take a food protein, you can isolate branches of gluten, right? So you get more specific antibody response. You can do the same thing with foods. You can just measure the lectin component of food on a well, and then you might miss it if you check the whole pea protein, but you may catch it if you just check pea lectin. So for lectin-based foods, they check the lectin and the whole food protein. And then for food, they check cook and raw. Like this is the ultimate model of doing testing. So this is the Cyrex Array 10 profile, and they looked at cooked versus raw, and then they check different types of lectins, and specific oils have specific properties as well. So you can see pea protein, pea lectin in the bottom. And they do that for the foods that are high in lectins. And if you don't have those specific measurements done, you can miss the reactivity of those types of reactions. Okay. Everybody good so far with that? Okay. Now, one of the things that I also want to share with you, and we're going to talk about this more when we talk about how to work up an autoimmune disease patient. I'll do like an array 10 for a couple of reasons for some people. I want to see, have they lost total immune tolerance? Okay. So if you take a look at this patient, do you see a lot of antibody reactions? No. So if I saw this, I'd go, you know what? Their immune tolerance to food proteins is pretty good because they have an autoimmune disease, but they only have like one slight reaction. But if I saw a panel where almost everything was reactive, like let's say everything was, like, this isn't even that bad, but let's say everything was in the yellow or red, I go, oh my God, they're testing to every single food protein. And in that case, if you see a majority or half or almost 40% or something, a third, most of the food protein, a lot of food proteins reacting, you have to just kind of go, what are you going to do? Because you can't remove every food protein. Okay? So then you have to focus on working on their oral tolerance, and then the test becomes a pre and post. So I'll give you an example. If I get a panel back, first, well, let me go even further. In my practice, if I have a patient come in that has autoimmune disease, if I'm going to stick with the general guideline, I'll stick with an autoimmune paleo diet. If I'm not sure, like just avoid lectins, avoid nightshades, go off gluten and dairy, and make sure your diet's very, very diverse. That's the least antigenic food protein diet. Because nightshades are very inflammatory, lectins are very inflammatory because of the protein structure, gluten has new wheat proteins, and then many people react to the new wheat protein and then cross-react with many grains. And most patients have noticed those observations too. I remember before the autoimmune paleo diet was a name, I would have patients come in with Tupperware of their own foods because they couldn't eat anything. They had to come into the office and have snacks, but they had to cook it themselves, had to prep it themselves, and they only ate about 6 things because they were reacting to everything. We didn't call it autoimmune paleo then, it was just what we called it. And we noticed that most patients that were really sick were basically just eating veggies and meats. And they couldn't handle sugar spikes, they couldn't handle any kind of grains, they couldn't handle cheeses, you know. They went to the whole thing of like, whoa, I can't eat cow's milk compared to goat milk, and they realize they can't do goat milk either, and they finally go down, and then they eventually end up with no milk, or maybe almond milk, or maybe not even that. I mean, they're all different, right? So you see these diets kind of change. So when you see people that test for every single food protein, then you know that they've lost their immune tolerance. And then my clinical strategy isn't to get them off every single one of those foods. So if you get them off all those single foods, where you test, all the new foods they're eating are going to now show up because their immune system is reacting to those. So we'll do the first test as their baseline and just work on their immune tolerance, which we'll talk about tomorrow, and then come back and see if those food proteins change. The amount of food proteins, the degree. If they had 60, half the test was panels, now they're down to 20%, I know their tolerance is getting better. Now, if I have the autoimmune, like if I have a Hashimoto's patient and I have a rate 10, I'm going to compare it to the study we published between what their antibodies were that are elevated to what we found cross-react. So if I see scallops high and I look at the list and I see scallops did cross-react with T4 and they have Hashimoto's, I'm going to say no scallops for you. So if I do have the privilege of looking for cross-reactivity, I will. For the most part, most autoimmune diseases, the only cross-reactive foods are gluten and dairy. That's why they're so critical. Like with RA, MS, type 1 diabetes, gluten and dairy are cross-reactive. Okay? That's why when you see those reactions, you definitely want to get them off the food, because cross-reactive food reactions are devastating for people because of the amino acid similarity. Okay? Now, if I don't have the cross-reactive list and I do a rate 10, because they have an autoimmune disease like MS, and we don't know what all the key factors are besides gluten and dairy that cross-react, I ask him, like, do you notice a reaction to any of these foods you tested positive for? If they go, yeah, I kind of suspect when I eat melons, I don't feel well. Okay, well then avoid melons and then eat everything else. And then I'll work on their tolerance, because you can't do a repeat test for tolerance unless they're still eating the same foods to see if their tolerance is getting better. You have to have them actually eat the same food still. And while they're eating those foods, see if they're less reactive to them. Does that make sense with everyone? Okay? Let me teach it all over again, starting from scratch, in a different way. Okay, you guys ready? Let me think about how I'm gonna do it. Okay, here we go. You're a clinician. Patient's gonna come in with autoimmune disease. You're gonna have to go, what am I gonna do with their diet? In a real setting, you're gonna have to evaluate your patient's motivation to change and decide how deep you wanna go and how deep they can go. Maybe they're not ready to go. Maybe they're really motivated, but it's hard for them. Maybe they're really scared. Maybe they'll tell you, like, I can go piece by piece, but I'm not ready to commit to everything yet. I just need to work through it slowly. I don't want to do a lot of drastic change. If I do a lot of drastic change, I feel sick sometimes. I just want to go slowly. Okay, maybe that's your scenario. Maybe the other scenario is, I'm suffering. I will do whatever it takes. I don't care about anything I eat. Just tell me what to do. Different scenario. Give a third scenario. Patient comes in and goes, I'm already on autoimmune paleo diet. I feel horrible. What do I do now? Different scenario. Make sense? So you can have all these different scenarios with different types of motivation. Let's talk about the unmotivated patient first. Hard to change. For me, I would always start with gluten dairy free. Go gluten dairy free. And if they can handle gluten dairy free, and actually I'll show you this chart. Here we go. It's actually an order of steps. Go gluten dairy free first, then try paleo, which is grain free, and then the deeper level will be autoimmune paleo. Or you can sit down with your patient and go, listen, We can test for foods if you want. We can go straight into a diet. Let's assume we're with a patient who just got diagnosed with autoimmune disease and they want to do some food interventions. There's different degrees of levels of intensity you can go. You can just go gluten-free, which would include dairy-free, gluten-dairy-free. You can go paleo, which is also grain-free, or you can go autoimmune paleo, which is lectin and nightshade-free. And they may say, I feel awful. I'll just go autoimmune paleo. I don't care. Then you go, great. That's your choice. Next person goes, well, I really like nightshades. I really like tomatoes. I don't know if I really react to it. All right, we'll try gluten-dairy-free first. If you're not getting better, we can go further. There's nothing wrong with presenting the patient the options and have them think about it and go, well, let's try that. If it doesn't work, we can go deeper. And you can print out a diagram like this or do whatever you want with your own diagrams, but it helps patients understand what's going on. Okay? So for the person who's jumping into it, you can test. You can go into those things that way. If you have a person who is highly motivated, you have a highly motivated patient, you should always just jump into autoimmune paleo because that's where you see the greatest effect. And especially if you have an acute flare-up, like someone's on fire, they're barely getting through the day, just take away the most inflammatory food proteins, which are lectins, nightshades, gluten, dairy, and jump right into autoimmune paleo. Okay? Now, you have to make sure if they jump into paleo or autoimmune paleo, they support oral tolerance. And the key concept of that is they have diversity in their diet. Plant fiber diversity. You go, okay, you go autoimmune paleo, but you can't have the same meal every day. Can't have the same 4 vegetables. You really have to diversify. We'll talk about strategies how to diversify that further Sunday, okay? But you really want to have a diverse diet. And that's the other thing you'll see with people that come in with autoimmune diets. They'll come in, they're autoimmune paleo, they've limited their diet, they're not eating much of any diverse types of vegetable diets, and they're continually getting more and more food reactions. Lack of diversity means lack of microbiome diversity. Lack of microbiome diversity means more autoimmune flare-ups. So you kind of want to get a feel of what your patient's at and see what they're doing. I actually like to ask my patients, what do you have typically for lunch, breakfast, and dinner? Almost autoimmune disease patients, almost all of them, eat the same thing every day. Because they took it so long to find out which foods cause reactions, they're just thinking of not reacting. And they're not thinking about diversifying. So for a lot of autoimmune disease patients, your goal would be they're already on autoimmune paleo, and you got to teach them how to diversify. If you happen to have a type 1 or thyroid patient, go deeper. Go to cross-reactive foods. Do an rate 10. Look at the research we published in type 1 diabetes and thyroid, which is in your notes in the paper sections, research papers. Look at that list, look at a rate 10, and find the foods that are on both, the study, and then that they have antibodies for, and get rid of those. Those are cross-reactive. You don't want those in their diet, okay? And don't forget about blood sugar stability, and don't forget about salt intake. Those are the key things with diet. Everybody good? Let's take a break, and then we'll get into infections.